Assalamualaikum and hi Saraiman. Today me Putri Nur Najwani binti Nur Asfizan and my friend Sarah Suraya binti Sherhan and Nur Anisafia binti Abdul Karim will present about Rhesus blood group system. So before we enter to the detail, we go for the table of content. First, we have introduction, 3RH nomenclatures, which is fisheries, Wiener and Rosenfeld, phenotype and genotype, Wikipedia, case study, also the conclusion. Introduction to RH blood group system. So before we enter what is the RH blood group system, we must know about the origin of the name first. The term rhesus factor comes from early experiments using red blood cells from rhesus monkeys to obtain the first blood serum. So, what is the RH blood group system? It is a human blood group system with proteins on red blood cell surface. It also plays a crucial role in blood transfusions and pregnancy-related complications. So, why is it important? Because that is the second most important after the ABO blood group system. It's also commonly involved in transfusion reactions. Key facts about the RH system have three. First, we have the number of antigens. So we have 49 antigens identified in 2005 and over 15 antigens as of 2023. For the key antigens, we have big D, big C, big E, and small e. There was no small d antigen. For the Rh-D status, we have two, Rh positive and Rh negative. So for the Rh positive, we have the Rhd antigen. Example, A positive equal to A antigen plus Rhd antigen. For the Rh negative, it lacks the Rhd antigen. Example, A negative equal to A antigen but no Rhd antigen.
So now I'm gonna be explaining on Rhesus nomenclature. So what is Rhesus nomenclature? It is a common language for identifying Rhesus antigens and determining compatibility for transfusions and organ transplants. So there are three Rhesus nomenclature that I'm going to be explaining in this presentation. So the first one is fisheries, the second one is Wiener, and the third one is Rosenfeld. So now we are going to start with Fisher-Race nomenclature. So Fisher-Race nomenclature contains three pair of genes that are responsible for the expression of rhesus antigens on the surface of red blood cells. So DCE, which is three loci, determine the presence or absence of specific antigens. So as we can see here in this picture, the D is the main antigen and its presence is the most important in determining in rhesus blood grouping. Meanwhile, C and E are expressed, but they are less critical than D. So Fisher and Race named the antigens of the system D, C, C, and E. So D, little d is an amorph, which is a gene with no antigenic product, meaning it is silent unless D is absent, and the letter D is used as a placeholder. Also, according to Fisher's theory, an individual who inherits a set of rhesus genes from each parent, either one big D or one small d, one big C or one small c, one big E or one small e. So this combination of genes inherited will be called halotype. So as we can see here, based on this example, so the mom has DCE, meanwhile the dad has DCE, and all of them are small. So the child's genotype will be DCE/DCE. Okay, so this means their RBC will express the DC and E antigen. Since phenotyping detects the antigen present on RBC, this person's phenotype will be rhesus positive, D positive, because they have at least one D antigen. So next, we're going to be discussing on Wiener nomenclature. So Wiener proposed that the Rhesus blood group system is actually controlled by two genes, one inherited from each parent. So each of these genes is responsible for producing a single structure on the surface of RBC called an agglutinogen. So an agglutinogen is not actually a single antigen. It actually consists of three smaller antigenic components called epitopes, as we can see in this picture here. So this epitope can actually react with specific antibodies, antisera during blood typing tests, which help us to identify a person's rhesus type. So there are three egg major agglutinogens as we can see here. So next, we're going to be discussing on the comparison of Fisher-Race and Wiener. So actually, immunohematologists, which is a blood specialist, they are often use a combination of both systems to record blood types. So Fisher-Race notation is commonly used in discussion of genetics. Meanwhile, Wiener notation is useful for simplified shorthand and for identifying halotypes on chromosome. So as we can see in this table here that I provided, so as we observe, both systems describe the same rhesus antigens, but actually in different ways. So how can we identify? You must be able to convert between Fisher-Race and Wiener notation. And you also need to understand these notations to help with blood transfusion compatibility and risk typing. So next, we're going to be discussing on Rosenfield nomenclature. So Rosenfield nomenclature actually demonstrates the presence or absence of rhesus antigens on RBC. So it assigns a number of each antigen in the rhesus blood group system. So how it works? So each antigen, D, C, E, and small c and e is given a numeric value. As example here, D is called Rhesus 1, C Rhesus 2, E Rhesus 3, small c Rhesus 4, and small e Rhesus 5. So minus sign indicates absence of the antigen. For example, Rhesus 1, 2, 3, negative 4, negative 5 indicates D positive, C positive, E positive, C negative, and E negative. So if an antigen is not tested, it is not included in the sequence. So what are the advantages and limitations for Rosenfield nomenclature? So the advantages is concise phenotype description. It simplifies the documentation of RBC phenotypes, and also the numeric system is compatible with electronic systems, which aid in data entry in retrieval. And the next one is limitations. Similar nomenclature exists for other blood groups, which is Kelly, Duffy, and Kidd. So both the alpha designation on races and the numeric code 1 to 3 etc. must be used to avoid confusion. And next, I'm going to be discussing on phenotype and genotype. So here I made a table comparison between phenotype and genotype so you can get a clear view and enhance your understanding more on the difference between phenotype and genotype. So what is phenotype? It is a result of reaction between RBC and antisera. Meanwhile, genotype is genetic makeup predicted from phenotype and race. So what is the determination method between phenotype and genotype? So for phenotype, it is based on agglutination reaction with antisera, which is positive or negative. Meanwhile, genotype, it is predicted using statistical probability and family studies. So for a key example, we can see here DCE, small c and E antigens on RBC surface. And also for genotype, it is actually rhesus 0, DCE, rhesus 1, DC, and small e, and as we can see. As for testing requirements, use of NTD routine testing other antisera for specific issues. Meanwhile, for genotype, molecular testing or family studies to determine true genotype. And what is the use for phenotype in clinical practice? It is for parental testing, hemolytic disease prediction, and Rhesus antibody specificity. Meanwhile, genotype is for genotype prediction, fetal Rhesus testing, and rare antigen detection. For limitations, phenotype cannot determine true genetic makeup, only phenotype. Meanwhile, the limitations in genotype require statistical prediction or family study for accuracy.
So next is weak expression of D. So what is weak D antigen? It is a phenotype where the D antigen is weakly expressed on red blood cells. It's not detectable by routine methods, which is spin tube technique. So how is it detected? First, use of prolonged incubation at 37 Celsius and use of anti-human globulin tests. So for the key characteristics, it usually has D antigen but in fewer numbers than normal Rhd positive cells. It also reacts with some anti-D antibodies but not at all. And, and individuals with weak D cannot produce anti-D antibodies. For the transfusion consideration, it's usually treated as Rhd positive for most transfusion purposes. So we have three mechanisms are responsible for the weak D. First is genetic weak D, second is C-trans, third is partial D, or we can call as D mosaic. So for the first type is genetic inheritance of weak-D. So what is genetic weak-D? It is a condition where the D antigen is fully present but in smaller amounts on red blood cells. It's caused by variations or mutations in the Rhd gene. So who has it? Found in less than 2% of Caucasians and more common in African Americans. For the key feature, the antigen is complete but appears in lower numbers on red blood cells. So, for the second type is C-transweak D or position effect. So, what is C-transweak D? It is a type of weak D caused by the position of genes. It's not a mutation. It also occurs when the D gene is on one chromosome and the C gene is on the opposite chromosome, which is transposition. So for the genetic example, we have big D, small c, and E, and small d, big E, big C, and small e. So the big C gene is on the opposite chromosome from the big D. So effect on the big D antigen. So the big C gene in transposition weakens the expression of the big D antigen, and the big D antigen is complete but appears weaker in tests. So for the clinical significance, people with C-trans weak D are considered the Rhd positive for transfusions. So for the third type is partial D or D mosaic. So what is partial D? It is a type of weak D where some parts epitopes of the D antigen are missing or altered. Unlike C trans weak D, this is caused by mutations in the Rhd gene. So how is it different from normal D? So some D antigens parts are missing or changed. Also, the D antigen appears weak or incomplete in testing. So for those people with partial D can make anti-D antibodies if exposed to normal Rhd positive blood, example through transfusion or pregnancy. For the clinical significance, some partial D individuals are treated as Rhd negative to avoid transfusion reactions. It's also important in pregnancy to prevent hemolytic disease of the newborn. So, Rh immunoglobulin and weak D status in newborn. So for the situation, mother has Rhd negative, means that does not have the D antigen. Baby have weak D positive, means has a weak expression of the D antigen. So why is this important? So if fetal red blood cells, weak D positive, enter the mother's bloodstream, her immune system may recognize them as a foreign and produce anti-D antibodies. So these antibodies can attack the red blood cells of a future Rhd positive baby, causing hemolytic disease of the newborn. So is Rh immunoglobulin needed? Yes, Rh immunoglobulin is usually given to prevent the mother from making anti-D antibodies. However, some policies may not require Rh immunoglobulin if the baby has a confirmed weak D type that does not produce anti-D. So last is when should weekly testing be done? So we have three situations where weekly testing may be needed. First is from blood transfusion recipients. It's usually to ensure they receive the correct RH type. So second is expectant mothers. So it helps determine if RH immunoglobulin is needed to prevent complications in pregnancy. Third is newborns. If the mother is Rhd negative, testing the baby helps decide if Rh immunoglobulin is required. So when weekly testing is required is from blood donors. So it's to correctly label donated blood as Rhd positive or Rhd negative.
Now we move to the case study. The topic of the case study that we pick is blood incompatibility in a patient with rare Rhesus phenotype. The case overview. This case involves a 39-year-old woman from Bam, Kerman, Iran with ovarian cancer who developed anemia and required a blood transfusion. A blood transfusion became complicated due to a complex history of severe chronic hemolytic anemia, difficulty in finding compatible blood due to allo antibody production, and the rarity of her blood group. The patient's blood was tested extensively for compatibility, and phenotypic evaluation of her family members were performed to identify potential blood donors. These are all the patient history. The patient is female in 39 years old. The medical history shown diagnosed with ovarian cancer, and she has severe chronic hemolytic anemia with hemoglobin levels dropping to 5 to 5 g per dl. She also has history of transfusion reactions characterized by fever, chills, and hemolysis. She has blood compatibility issues where she experienced incompatible cross-match with all blood units in the hospital with 3 plus reactions. She has previously performed base blood transfusion based on ABO and Rhesus compatibility without RBC phenotyping, leading to stimulation of the immune system and production of allo antibodies. The blood type of the patient is O plus based on the reverse typing method. These are the lab diagnoses that has been done in the hospital. First, we have initial antibody screening test. Antibody screening direct Coombs test and antibody identification tests were performed. The presence of both alloantibodies and autoantibodies in patient serum. Autoantibodies interfere with the reliability of results. The second test, we done repeated antibody screening test. A 3-cell antibody screening test detected alloantibodies. Di mana antibody pesakit akan attack antigen daripada donor. Antibody identification reveal specific antibodies for rhesus antigens, which are capital E, capital C, and small c, with 3 plus reaction. And next, kita buat juga RBC phenotyping test, di mana the patient's red cells were phenotyped as D positive, capital C negative, small c negative, capital E negative, small e negative, K negative, FYA negative, FYB negative, M positive, N negative, capital S positive, small s positive, and JKA positive. All the above tests were performed in duplicate to confirmation of results. Mana kita buat sebanyak 2 kali lah untuk confirmation. And next, yang ni adalah yang paling utama, yang mana kita buat family phenotypic evaluation. The results of phenotyping for family members were documented as below, showing that two brothers had matching phenotypes with the patient. Kat sini kita boleh nampak brother number 2 and brother number 4 sama. Dia punya blood group, dia punya rhesus dengan patient. Yang mana ini akan menjadikan the two brothers as a potential donor. For treatment, kita ada 2 treatment. And first, kita ada immunosuppressive therapy. Yang mana patient akan treated with immunosuppressive therapy for 4 months to manage the autoimmune response and allo antibody production. And the second one, kita ada further treatment. Yang mana patient memang memerlukan blood transfusion kan untuk anemia. And then kita akan teruskan dengan blood transfusion since the two brothers were identified as potential blood donors. Next, possible future solution. Yang mana donor ataupun recipient yang ada rare blood group, diorang akan buatlah solution-solution ni untuk for the future. First, family members with similar blood types should be encouraged to register with blood banks for emergency situations. Yang mana jika recipient memerlukan blood tu lagi, blood bank akan terus contact the family members untuk derma darah lah. Sebab itulah kena register supaya senang untuk contact in case ada emergency ke apa kan. And the second one is rare blood donors may consider cryopreservation of their units to ensure availability in emergency. Yang mana donor-donor ni kena freeze kan diorang punya RBC. Cryopreservation tu kita akan freezekan dia punya RBC dan dia punya stem cell semualah. Kita akan preserve dia untuk kegunaan in the future lah. Yang mana kat sini dia ada gambar dia ni. Yang ni dia akan freezekan. Sebelum tu kita akan pakai cryoprotectant untuk menghalang daripada RBC tu damage sebabkan oleh freezing tadi tu. Yang mana biasa kita akan guna glycerol lah untuk protect the RBC tu. Sama juga macam kalau orang buat cord blood punya preservation kan, macam tu, lebih kurang macam tu juga. Tapi yang ni kita akan simpan blood tu lah, kita akan freeze blood tu. And the third one is if the patient is not anemic, autologous blood donation could be another option with freezing for future transfusion. Yang mana jika patient yang ada rare blood group ni tak ada anemia ataupun tak anemic, dia akan dermakan darah dia sendiri dan kita akan freezekan dia punya darah in case in future patient tu memerlukan balik darah tu, so kita akan terus transfuse lah dalam dia punya badan tanpa perlu contact donor ataupun cari donor ke apa kan. Maknanya dia akan guna balik darah dia tu lagi. To sum up the whole slides, the Rhesus system is a complex and essential blood group system that extends beyond the Rhesus D antigen. While Rhesus D is the most clinically significant, other Rhesus antigens such as capital C, small c, capital E, and small e also play crucial roles in transfusions, compatibility, and immune responses. Understanding the full spectrum of Rhesus antigens is vital in preventing hemolytic reactions, ensuring safe blood transfusions, and managing conditions like hemolytic disease of the newborn, HDN. These are all the references that we use to make this slide presentation. And thank you. That's all from us. Thank you.